So let's jump right into some introductory content. A lot of this should be review. Some of you will feel totally comfortable with this material. Some of you will be a little bit annoyed at having to learn this again. I'm hoping if it didn't make sense for you the first time around that we can help it make sense for you this time. This is really the foundational stuff that the course is going to depend on for the first few weeks. So if you didn't get this to make sense for you, please give it a shot. Ask me if you think you can figure out a way to get ahead and make sense of this. All right, so the topic is very simply genetic inheritance, but really the beginning of genetic inheritance. Of course, a lot of the semester will be talking about this. So helping us to answer some questions about why kids look like their parents. All right, boink, there we go. So a very basic, super basic question in biology, basic question in life, right? Where did you come from? That's one of the reasons I like genetics. We can answer this question, not holistically. I can't tell you everything about yourself, but I can tell you, or maybe show you some of the tools that could be used to tell you about yourself. So where did you come from? Usually open this up to the class. Where did you come from? All right, babies, parents, birds and the bees, right? So some contribution from your mom, some contribution from your dad came together to make you. And people, even without a basic knowledge of genetics or even a concept of genetics, could probably take kids and point them to their parents and say, okay, I understand this kid is from that parent, right? So we have kids that have features that make them generally look more or less like their parents, and a lot of that is down to genetics. So you first became you on the path to being you when a sperm cell from your dad got together with an egg cell from your mom, and the fancy term for those cells are gametes. So this is an image of an egg cell, the big oval shape here. Egg is one of the gametes that's produced by the female. And then also shown in here, a bunch of sperm cells. In humans and mammals, sperm approach the egg lots at a time. And so the chance that any one sperm gets into the egg cell, fuses with the egg cell, is pretty low for an individual sperm cell, but fertilization actually only happens when one sperm cell gets together with that one egg cell. In spite of there being lots of sperm cells, they don't all make it. So these are the gametes, and these are produced by the adult parent individuals. So a male is producing sperm gametes, and a female would be producing the egg gametes in this arrangement. So what's inside of there? One of the things I think is fascinating, and although we probably know it, I don't know if you really thought carefully about it, that there was one cell, and in that cell, whatever about you is genetic, was in that cell that formed when sperm met egg. They fused together and they brought that material, and you were already at one cell, you were on your way to having your eye color, your hair color, programmed parts about your height, your dietary preferences, all kinds of stuff was programmed into you at a teeny tiny one cell stage. So the programming comes down to the material that's on these chromosomes here. We'll talk later about what is a chromosome, and much later, I guess, in the molecular biology portion of genetics, we'll be talking about really the molecules that make up chromosomes. For now, we'll just talk about them as these shapes, and suffice it to say that humans have 23 kinds of these. So 23 pairs of chromosomes in an adult. And we're starting with the gamete here. So one of these cells, the sperm cell, fused with the egg cell, and the sperm cell then would have half of 23 pairs, and the egg cell would have another half of 23 pairs, and they get together. So your egg cell has a version of chromosome 1, and your sperm cell would have a version of chromosome 1 all the way through. So one version per gamete, and then the gamete fusion is the process of fertilization that put those together to become you. As an adult, now you have, as a normal adult, you would have 2 of each kind of chromosome with these different numbers. So 2 of chromosome 1, 2 of chromosome 2, and so on. So they get these numbers by convention basically decreasing in size. So this is just a cartoon version, but you can see number 1 is rather large, making it down to chromosome 22, not very large, and chromosome 23 is a little special. We'll talk about that later. So we got our gametes, and gamete fusion produced the first cell that became you. That was called a zygote. And as you grew up, you have lots and lots of cells, and your cells now are known as somatic cells, referring to soma, or the body. So the body cells, most of your cells don't participate further in sexual reproduction. So your finger cells are not responsible for transmitting any genes to your offspring. Those genes that are going to go to your offspring, should you have any, are contained within your what we call germline cells, so special cells in the gonads of your body that are preparing to distribute that genetic information to next generation. Okay, so we got 23 pairs now, and the pairs, you can see the 2 kinds of chromosome are the same size and shape. We call that a homologous pair. And although they are paired up nicely in these diagrams, in the cell itself, you wouldn't necessarily see these chromosomes being friendly with each other. They are more or less independent. They do their own thing. We'll see as they do cell division that these chromosomes go into cells independent of each other during the process of mitosis. And we'll get into that in much more detail later. But these are the chromosomes that you could find in a cell. They also wouldn't necessarily look like this. This is a nice handy diagram showing, just showing what's in there, but not necessarily how it looks or where in the cell it would be located. Got to simplify things here and there. Okay, so this is a little more realistic, away from the cartoony version. This is what we call a chromosome squash. You're actually in the lab going to do a chromosome squash on fruit flies. And this technique allows a person in the lab to stain the chromosomes with a dye that allows them to be seen. And then by pressing very hard down onto this preparation, this pushes the chromosomes apart so that we can see them as distinct units. Chromosomes appear like this for only a little bit of the cell cycle. So this is chromosomes packaged up in preparation for probably in this case, mitosis. So cell division, the chromosomes get packaged up into a way that we can see them distinct from each other. And usually I do this right. This is the chromosome set for a human female. Let's see how many we can count. I'm going to count them by the dark centers that appear to be staining more darkly at the centromere. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40. 1, 2, 3, 4, 5, 6. Yes! Good as ever. So 46 chromosomes are what you would count. If you got into the nitty-gritty of this, you would be able to find them and pair them up as 2 that look the same, have the same shape, same centromere location, and those would be the paired chromosomes. So 23 pairs gives us 46. All right, so we know what's in a gamete. We know what's in a typical body cell. How do we get from the body cell to the gamete? The body cell then has twice as many chromosomes as the gamete. How do we get there? That's a process we will see a little bit later, but we'll just give it a name for now. That's the process of meiosis. And meiosis involves going from the typical chromosome complement in your body to this half amount complement. So down here we would see 46 chromosomes in pairs in the body cell. And then after the process of meiosis, good one to know the name for now, we'll talk about the details of that later. After the process of meiosis, cells are formed that have half that amount. So this is 23. So to go from 46 to 23, we call this the diploid number. 2n is a diploid number. And then we move over to haploid or n number. So basically as human cells, whether you're talking about a somatic cell or a gamete, that number, the base number is the same. We just have the chromosomes paired or not paired. So the life cycle is this process where we go from diploid to haploid and then back to diploid. It differs among organisms. In humans, diploid is most of our life, right? So from fertilization all the way up until you happen to make gametes and produce another generation of humans. That's this portion down here, the diploid portion of the life cycle, and then very brief portion, hours and days of the gametes being in existence and then combining back together. So full double amount of chromosomes, half amount, and if we're going to get back to double, half has to combine with another half. So egg has half, sperm has half, they meet together, process of fertilization, that gets us the back to the diploid amount, and then this cycle continues over and over and over again. Okay, so chromosomes, as I mentioned, they get their distinct numbers. These are numbered by convention, so we know that humans have 23 kinds of chromosomes. They get numbered from biggest to smallest, more or less, and so they would have a name, something like chromosome 1. And chromosome 1 in humans should have the genes that are usually found on chromosome 1. So if we're finding a gene, if we happen to locate a gene that's responsible for a phenotype, then we could find out which chromosome it's on. When you guys do your fruit fly labs, you're going to find that you have a gene that you're studying, and that gene has a location on one of the fruit fly chromosomes. And that's something that's pretty stable throughout the species. And because we have diploids, we have paired chromosomes for each of these, 1, 2, 3, etc. And the chromosomes in the pair, we call those a homologous pair. So this term homologous refers to them being the same size with the same centromere position. So the centromere is shown here as just a little pinched portion. This will have a function in mitosis and meiosis, we'll see later. But notice these two chromosomes are about the same length, but they have different centromere positions. So we could tell them apart potentially by that feature. So chromosome 1, chromosome 2, chromosome 3, etc. And everybody gets a homologous buddy. If you're wondering about the coloring is sometimes used as it is here to indicate one chromosome from mom and one chromosome from dad. So when you have this homologous pair, you've got a dad chromosome and a mom chromosome for all of your 23 pairs. All right, so we're going to get into genes and talk about genes and alleles for a while first, before we get into much detail about chromosomes. And so if you're studying genes, you would be studying the alleles of those genes. So alleles are different versions of the genes. And so they can actually make different gene products, different proteins, create different phenotypes, etc. So this B gene here, the B gene has a gene located on dad's version of chromosome 1 and a version located on mom's copy of chromosome 1, and they happen to be different. So dad gave us the big B and mom gave us the little b, but those genes are located at the same place. This is another feature of homologous chromosomes, is that they should have the same genes at the same place, same position along the chromosome. Down here we have the T gene, which is lower t, little t allele for both chromosome, homologous chromosomes of the pair. Here's the Q gene. Now dad gives us a little q and mom gave us a big Q. N gene, everybody gets a big N, and so on. What I'd like to highlight with this illustration also is that there are always thousands of genes under the surface. If you're doing a genetic study, you're typically trying to focus on a few of these. We can't think about 40,000 genes all at the same time, so we sort of parcel it out and deal with what we can at a time. So in this example, we might be looking at 4 genes, 2 alleles each, and trying to figure out what contribution these might have to the phenotype we're studying. So there's sort of this background amount of genetic variation that we may not be looking at. We'll look at one thing at a time. Okay, so looking back, kind of combining our chromosomes with the life cycle, one of the big questions of transmission genetics is, what are the chances that a parent gives some genetic information to their offspring, right? So if your mom has an eye color that you're interested in, what are the chances that you would get that eye color as her child? And that comes down to the ability to transmit the alleles for what your parents have, and also this feature of dominance and recessiveness that we'll talk about later. So let's say we have a diploid individual and we're looking at genes that are located on this chromosome, and the parent here has a big G or a little g for the gene we're looking at. So during the process of meiosis, only one of those gets into a gamete. Which one? Good question. The one that gets into the gamete is up to random chance. So we have two, sort of like flipping a coin. Every time a gamete is made, it's one or the other of those. So basically for us, this is going to be equal chance. So if there's a big G chromosome and a little G chromosome and only one of those gets in, then it's going to be half of the time little g, half of the time big G. Right? So in this illustration, we'll just go ahead and say that the big G made it into the egg cell, which is shown here. And if we wanted to complete the diagram for this egg cell to go on to produce a somatic cell or an individual that is heterozygous with two copies, two kinds, big G and little g, then the little g would have to come, in this case, from the sperm cell. So big G and little g can combine together to give us this big G, little g combination in the grown-up. All right, so two kinds, half chance each will get in. Here's another one, same thing, different gene, different chromosome, also heterozygous. Heterozygous means that it's got two different kinds of alleles. So the D gene now has the big D allele, the little d allele, and there'd be a half chance that little d ends up in the gamete. All right, not too hard. Things, of course, get a little bit more complicated. So let's say these genes belong to the same individual. This person has G genes and D genes. What are the chances that a person who's heterozygous for G and heterozygous for D contributes both a little d and a big G to their offspring? So this gets a little bit more tricky mathematically. Basically, so there's a 50% chance, as we've seen, of getting big G and a 50% chance of getting little d. But if we combine those chances together, those are one outcome out of four possibles. And I'll show you some ways to think about that coming up. But basically, if you've got D's and G's and two kinds of each, then you could get big D with big G, big D with little g with big G, little d with little g. Let's hope I did that right. So 1 out of 4 chance here, or a half of a half is another way to think about that. All right, and you can do this a little bit more complicated. Let's say we have 3 genes and we're interested in calculating how these end up in a gamete. So we've got heterozygous for B, heterozygous for Q, and homozygous for N. So homozygous, only one allele kind for that N gene. So if we look at the chromosomes, these chromosomes would belong to an individual with the genotype that's shown here. This is probably something you've covered before, this kind of genotype notation. So big B, big Q, big N, big N. The alleles for the genes are paired. Typically, by convention, we put the capital letter first, and the order of those genes sort of up to whatever you're doing, but it's good to be consistent. So I've got them not alphabetical order, but just going 1, 2, 3 across the chromosomes. We're going to introduce a notation in this class. Sometimes it's going to be more useful for us to use this notation. Here we have B's separated by a slash. That means 2 alleles on homologous chromosomes. The semicolon then separates out the next chromosome. So B/B, semicolon, then the genes on chromosome 2, big Q/little q, semicolon, genes on chromosome 3, big N/big N. So we'll use that a little bit more as we go on. Get familiar with that. Okay, so let's say we have 2 kinds of B, 2 kinds of Q, 1 kind of N. How do we combine those? There are a couple different ways. This is one way where you can use this so-called branch diagram, and you're basically just dividing up the possibilities for as many genes as you have to. So we're going to do this one gene at a time. For the B gene, we had a big B or little b, equal chance of those. If you've got a big B, that's sort of independent of the little b situation. If you've got a big B, there's still a half chance that you would get a big Q or a little q. Same thing if you got a little b, half chance you got a big Q or a little q. And so these end up breaking those halves in half again. Now the N gene becomes a little bit easier here. There's only one kind of N. So here we have big N, 100%, 100%, 100%. So we've cut this half possibility into 4 possibilities, and then it stays at 4 possibilities through the N. And finally, these are the resulting genotypes we would expect in the gametes. So big B, big Q, big N results from following this track. Big B, little q, big N results from following that track, and so on. So we get 4 outcomes, and mathematically, these are equally likely. So there's nothing controlling big B going with big Q or anything like that. They're independent outcomes. And therefore, the outcomes that are 4 different possibilities are all going to happen same chance. Another way to do this, sometimes I hesitate to introduce this, but because people, I think, can get this confused with the Punnett squares that we'll do later. But it's the same sort of mathematical trick, where if you want to combine independent outcomes that are equally likely, you can put them into one of these square diagrams and put them into combinations. So what we've done here is we have the B alleles here for gene 1, the Q alleles for gene 2, and we're going to combine those following B's down and Q's across into their combination. So big B meets with big Q, that's one possibility. Little b with big Q, that's one possibility. And so these are 4 possibilities shown here, all again equally likely. We haven't done N. Maybe you're already thinking your way through N. If you're going to do N, and you could take those 4 gametes that we solved for the 2 genes and then just add on the N gene. So the N gene here has only 1 allele, as we've seen. So there's not any other way to combine these. This is a homozygous gene, and it's going to combine with those B and Q alleles like so. This gets us the same result as we saw on the previous slides. 4 outcomes, all equally likely. So what we've done already is actually shown 2 of Gregor Mendel's laws. We'll talk about Mendel in our next segment, next presentation. Mendel came up with these laws, or these are laws, sort of laws of genetics, laws of introductory basic genetics. The first of those we call segregation of alleles. So we've already done these. I'm just going to put a name on them. And these are, I think, ones that are helpful to know the name for, and also helpful to be able to think about them and define them as you're setting up and solving genetics problems. So segregation of alleles, segregation refers to going into different places. And so for any one gene, its 2 alleles are going to have an equal chance of getting into gametes. So particularly if you have a heterozygous gene, there's an equal chance that big B will end up in a gamete or little b will end up in the gamete. Pretty easy, right? We've already seen that. Now I just have a name for it: segregation of alleles. The other Mendelian law that's worth knowing is called independent assortment. And this refers to 2 genes that are located on different chromosomes. So if you want to know how B alleles are going to go into a gamete relative to how Q alleles go into the gamete, this is the law for you. So independent assortment says that, let's say if you have big B, there's still an equal chance that you would get little q with big B or big Q with big B. I guess the alternative is that there's not any better chance that big B goes with big Q or big B goes with little q. There's no partnership between the blue chromosomes here or anything like that. All right, so get yourself familiar with those, what they mean, how they apply, and of course being able to tell them apart. Okay. So we'll have some practice problems for you. Here's one to think about. Let's say you had 4 chromosomes, 4 genes that we're looking at, and these alleles on them, how would you solve this as far as producing the gametes? How many unique kinds of gametes? I didn't really talk about unique before. Unique means that each gamete combination is the only version of that combination. Basically what I'd like you to do, I'm going to back up a little bit here, is as you're solving these, you could just as easily make 2 columns each with a big N, right? That might be tempting to do. Some people do this. I think they do a little bit kind of mindless work, and I'd like to save you from the mindless busy work that might cause you some extra work. So to do this calculation, you don't have to do 2 versions of big N. That would be redundant, and that would get you 2 versions of the same answers. So we did only 1 version of big N, and that got us just 1 column here. So we took the 4 existing possibilities and made a combination that got us still 4 possibilities. And these are all unique from each other, right? So no other gamete combination would have big B and big Q, and so on. So those are 4 unique possibilities. A version that would not be unique would be if we wrote big B, big Q, big N again. That would be not so unique. We like to be unique, right? All right, so I'll set up some practice problems for you to work through on these, and let me know if you have questions. And moving on, our next topic will be looking a little bit more detail about how these gametes would get to be combined together.